It's the left and right, and then that.
This is forward.
Yeah, and then
Oh, this is going to be put on a video.
is your laser.
Okay. I'm Kevin Ripp from AQUAFIX. They call me the Bugman. We culture bacteria. Whether it be bacillus or nitrifiers, we culture bacteria. We started in wastewater years ago, and then we got into ponds, and quickly when we got into ponds, we realized we had no idea what we were doing. So we took a few steps back and started to figure out how can we use the correct bacteria cultures in a pond to work in harmony with what's in that pond and reduce the nutrients. And then also, how can we use enzyme catalysts to degrade aquatic plants? Whether they be algae or aquatic plants, we use specific catalysts to degrade them. So we don't focus on killing anything. We focus on making it more susceptible to attack, or after an applicator kills it, to make it go away. An example of an enzyme is like Adolph's meat tenderizer. That comes from papaya, and that's a protease enzyme that degrades protein. That's why you put it on that steak to make it soft. Another example is when you bite into an apple and leave it on the counter for an hour, it turns brown. That's a peroxidase enzyme. Everything we make is non-hazardous, but anyone who comes up to our door thinks we got some really bad stuff in there. And if you got a small company, the best way to keep people out is put some stickers on your door. You know, that's Darwin, and we try to follow natural selection. We can't fight nature, it'll beat us every time. So we've got to really follow what nature's doing, try to speed up nature, try to help it. Whenever we try to fight it, like that lake down by the restaurant, it's really clear right now. I could put bacteria in there that'd make it even clearer. But in 4 months, it would kick back and then get some nasty form of algae that's been waiting to grow, that's been recessed, and it just comes to the surface. So all of our products are really, we try to follow the path of least resistance. There's some bacillus organisms under the microscope. We grow those, lots of different types. They have a good purpose. That's one of our labs. That is a working lab, and we use that to culture bacteria. 3 things. One, we bring in water from lakes, and we recreate that lake. And then after a period of 6 months, we will look in there to look and see what's the dominant bacteria in there. We can control the pH, the alkalinity, the temperature, everything about that lake. We can create in that vat. And then we can look at there after a period of 6 months and look at what the dominant bacteria is and use that information to help that lake owner. Now, we can't afford to do this one lake at a time, but what we do is we do this and we look and say, okay, these are really beneficial bacteria in this ecosystem, and these are bad ones. You know, these ones are not going to help the applicator. We also get some enzymes out of these batch. If you take one of these tanks, if we put the right organism in them, if we put a lipid in there, we will get a lipase out. Lipase is an enzyme. But we can create all kinds of different enzymes in a crude fashion using this kind of thing. But that's our lab. We do some other work with people. Like people will say, do you have a microbe that degrades, you know, they'll give us a waste, and then we will inoculate one of these tanks with the microbe and the waste. We'll leave it go for 6 months, and after 6 months, we will pull out the dominant microbes to see what's going. Oh, that's not right. That didn't show up very good. That's a top down of our big tank. This is a big tank we use to, we can take the strains out of here, take them into there and grow them up if we want them. This is a big brewery kettle, essentially. We can grow them up to very high concentrations, and the organisms grown in this vessel tend to be really good at nutrient removal in high BOD situations, and these ones tend to be good at nutrient removal in low BOD situations, a lot like the lake. That's on my office wall. This is the holy grail of enzymatic pathways driven by white rock fungus. White rock fungus makes your peroxidase enzymes, and these are the different pathways that we follow in our office. When we get a plant in, someone sends us spike brush, or they send us widgeon grass or something like that. We look at that and we take the cell wall of that plant, we take it apart to say, okay, here's what it's made of. How do we degrade that? how do we get it to degrade? Because applicators say, hey, I can kill anything, but I need it to degrade. I need to go away. So we start looking at the fundamental issues of how we can get rid of that. That's our lab. That is over at the University of Wisconsin in Madison. And in the back we have we have all these algae, and you'll see those. We use that. It gives us access to a lot of equipment, a lot of faculty. It's a wonderful wonderful resource. For algae, we work with a woman named Dr. Linda Graham. She's a phycologist, and she helps us understand some of these. This is Deborah. This is our microbiologist. This is Lingbia back there. She killed it without using chemicals. She's not telling me yet how she's doing it because she always thinks I'm a monkey with the results or something like that. But she's using enzymes to kill Lingbia. Lingbia is made of a glycoprotein, is what we call it. So we look at that cell wall, which is actually UV resistant and resistant to about everything. And we say, how can we penetrate that cell wall and get in there? And this young woman is known for being very brash, but very on the money. She's a great scientist. As an example, one of our water quality reports, she identifies the algae. In a perfect world, she doesn't like to treat anything until she's gotten a sample so she can tell exactly what it is. We run all these water quality parameters, mostly not so much for the customer, but for us. People come to us with oscillatory or Lingbia or Pithophora, and we can keep a database of what levels trigger those algae. There's our autoclave microbiologists are notoriously paranoid of contamination. Our hood. These are all the reagents necessary to grow algae. For years we'd bring in these algae and they'd die. We bring them in, and the easy way to kill algae is bring it into captivity. It dies. So we'd have to look up all these different micronutrients, and she has to add. She makes little baths for each one, and then every week they get a little cocktail that's just perfect for them and grows them. When we get something in and someone says, "Can you help us, make this go away?" We will test everything under the sun. We'll test the Renovate, the Habitat, SCI 62, hydrothol. Whatever we got, we will test it. So we don't limit it to one manufacturer. And we test everything, figure out how to make that aquatic plant go away or how to make that algae go away. There's the microscope. I'm not sure what she had on there, but she's looking over there. This is a Zola. We got this from John. John over here is one of our applicators in Northern California. He brings us unique challenges because he works with nothing but billionaires. He lives in the area of the world where the billionaires are kicked out, or they're kicked out by the millionaires are kicked out by the billionaires. And but his customers want no chemicals at all. They don't want any chemicals in their pond. So we have to reverse engineer everything to figure out how to get rid of it. This one, we brought it in the lab and that did it. It's dying. So we got to figure out why it grows so abundantly, and in the lab it's dying. We'll take and we'll add nitrogen to it and phosphorus and play around with those levels to get it to grow. There's some bladderwort. It came in as seeds. We're growing it. We don't have a solution for it yet, but we're starting to grow it, learning how to grow, learning what motivates it, and then we'll start dissecting it and taking it apart, looking at the cell wall. How can we break this down? Yeah, it actually came in with a sample of spike rush, and then they looked up the seeds and they said, hey, this is bladderwort. We've been looking for that. They put the bladderwort in, they started growing it. This is outside of our office here. These are cattails. The next couple of years, we're coming out with an enzyme that will degrade cattails and make them fall. Applicators will call us and say, hey, I got these cattails, I killed them, but they're standing there. They've been standing for the last year. Can you make them fall? So we're coming out with an enzyme that gets into the base that makes them fall, and we have these three bins we got from the farm store, and this one here is falling over. Here's cattails in water. They take them and they put them in there, and they put fungus in them and whatnot. They study them just to figure out how they degrade, what makes them degrade. Here's some coontail. Again, you guys are really good at getting rid of this. We have some natural ways to kind of control it too. We got to be tough. We got to be careful how we market them, or I'll go to jail. There's duckweed and there's bladderwort. If you want to naturally get rid of bladderwort, you got to use something like orange oil or something like that to pop the vacuoles and make it sink. The natural way to get rid of this or bladderwort, anything like that, is to make it sink. If you get its head under the surface, it'll drown. That's the natural way to help in your chemistry. Get its head under the surface, it'll drown. This is fungus. This is really prized. This is white rot fungus. She guards that thing like a hawk. Like, don't be touching that. This is some really good stuff. This is the stuff that will break down trees. When you're walking around in the forest, you see the fungus growing on the forest. That's the stuff. After the percent of water in a leaf or in a mulch gets below 50%, you'll start to grow fungus. And the fungus produces those enzymes that breaks down the lignin. And the lignin is the really stuff, the good stuff. That's where our stuff doesn't work. But quite honestly, on lignin, there's whole universities, there's whole wings that study the breakdown of lignin. I don't really know what that algae is. I just took a picture of it. I think it was red. I think she took that over to the university, and they were very excited to go looking for a sample of it. Java moss. There's more Lingia. That's what they call the glycoprotein. In a couple situations in Florida, we found that their phosphorus was high, but the nitrogen was low, so we started to lower the nitrogen, and the Lingia went away. We did that in a handful of situations this year. We also had a handful where it didn't work. So, but we're focusing on the nitrogen, and then we're working on a way to degrade that. We got all kinds of samples.
That was probably species differences.
What's that?
Probably species differences, the reason it didn't go away.
Okay.
We're probably dealing with different species.
Okay.
Having very different resistances,
Okay.
different requirements.
Okay. And that's why we get all the water quality data, try to figure it out. You can see we got this stuff all over the place. It's a big project for the next 6 months. They are Lingia. Yeah.
If you aerate or do anything different to that, can you force a retardation of its growth?
No, you know what grows this stuff like crazy? Anyone want to guess? We can add nitrogen, we can add phosphorus. If I want this to fill the tub in a week, what do I put in there? Sand. I put a cup of sand in there. Oh man, that woof, it grows. It loves that silica. It's a diatomaceous. Yeah, it loves it. It makes it grow. But that's kind of what we do when we bring it in. She puts it into vats. In this one, she puts 1 part per million ammonia. In this part, she puts a part per million of phosphate. This part, she puts sand. Trying to figure out, how does it grow? What makes this stuff grow?
Many ponds are silicon limited.
What's that?
I say many ponds are silicon limited.
Yeah.
It's very interesting that it grows, maximum growth if you add sand. That's
Yeah.
it.
You put the sand, and this stuff is tough. It loves to form that benthoic map over the bottom, and then it anaerobically takes the phosphorus out of the sand.
What if you wash that sand with acid?
I'll have to try that.
Try that.
I'll have to try
Because
that.
it may be something in the sand beyond the phosphorus doing that. Because you don't find that in silicon limited lakes.
Yeah. Yeah, that's right. That's like the pondyr nitrate. It's got silica. That's that. This is the stuff she killed. I don't know how she did it. She doesn't talk a lot, but she does work well.
Don't fire her yet.
Scott was up. We tried to get him to. We said, "Hey, we got this guy from Australia who wants to come in." She's like, "Nah, I don't visit him." Well, we tend to hire a lot of people who don't talk very much. Here's a maple leaf. Not a good picture, but you can see all the crap. Maple leaves are easy to degrade. Okay. Here's an oak leaf. That's been in that jar for 3 years, sitting in the sunlight. It's all full of lignin. So when someone says, "Well, I got a product that'll degrade oak leaf," yeah, and you're flying in on your Learjet and wind. You cannot degrade these. They're very tough to degrade. Whereas the maple, very, very easily degradable. Again, I guess that was oscillatory on the screen. Here's Phragmites. We're doing a lot of work with Phragmites. The chemistry of Phragmites is the same as the chemistry of the grass. I mean, the chemical makeup of the Phragmites is the same as the chemical makeup of the grass. So So people are getting a problem, they can kill them, but they got to get them to fall over. So we're working on getting these to fall over. We figure it'll be a 4 or 5 year project before we're done. Pithophora. I know you guys have seen this. It's one of our projects this next month, 6 months. If you look at Pithophora, what it does is it takes all the hardness in the water and all the iron and it protects itself in that. So essentially it's got an exoskeleton. The chemistry of the outside of Pithophora would be the same as what's on the outside of a crab shell or a lobster, anything like that. It's chitin. So it makes it very tough. Once it's going, it's very tough to penetrate. The only thing that will work is one of our enzymes with an acid-based algicide. If you use a high pH algicide, pH 8 or 9, it won't touch it. You got to get in there with an acid. But the harder the water, the tougher this is to get rid of. The harder and more iron, that kind of thing. Like it forms like an impenetrable exoskeleton. We have one in Florida. They have like 800 ppm of hardness. This is not going to go away. We're not going to have anything for Pithophora. There's another sample of it. I got all kinds of it. You can see we got all kinds of tests going on with it. It's baby pondweed. She's just working on growing that right now. Slender spike rush. If you can kill it and we can spray, we can stick bacteria to the surface, it will go away. One of the things this thing hates is it hates bacteria sticking to it. After it's weak, it hates bacteria sticking to it. We can really get it to go away. More of that. Spirogyra, very simple algae, but there's a reason we have this in there. We took a sample of Spirogyra maybe 3 years ago, took it back to the lab. We found 3,000 species of bacteria on here. We were able to identify 100 of them. So the question is, what are they doing there? They are breaking down food for the algae and feeding it to the algae. And we find that in a lot of different aquatic plants now. Aquatic plants and algae, we find that. There's a symbiotic relationship. The Spirogyra Pithophora cannot live without these special bacteria that they have producing food for them. I know what the food is, but I'm not going to tell you because if we figure it out, then we got something. We're trying to break this pathway down of all these algae that live on it. It's an amazing thing. It's like out in the Serengeti where you got this big cow and he cannot survive without flies on him. These algae cannot survive without these naturally occurring bacteria synthesizing vitamins for them. There's the water hyacinth and the duckweed. I got nothing for duckweed. Duckweed beats me every time, but someday we will. Here's all the different, algae there. Bill Cody probably knows the names of them. I've got them all right here if anyone wants to know. But one of the interesting things is we got 2 people in the lab now, and I said, teach them how to identify algae. And she said, no, I'll teach them how to identify bacteria. I'm like, well, teach them the algae. No. She said, algae is way too difficult to identify. I'm like, well, bacteria is difficult. But no, she's not as difficult as algae. There's so many forms, so many types. It adapts so quickly. This is Euglena, green in the morning, red at night. You can kill it when it's green, you can't kill it when it's red. Some people love it. If they have high BOD, a lot of sludge or muck in their pond, they will use something, they will get that. We have a product we put in the pellets. If we put that in there, the Euglena will go away because you're sucking up the BOD. More samples of the cell structure. We look at that a lot. We look at the chemical composition of it. I don't remember what that was. This is an early one we did with Cladophora. I know it's really easy, but our microbiologist said, no, we don't need to treat that with chemistry. I know how to do that. And she sprays this little enzyme on there right away. It's never come back 3 years later. So part of the key in our mind is not killing stuff, it's recycling it. Recycling it quicker so it just doesn't go to the bottom for the next go around. This is all parrot feather in the back. He couldn't get rid of the parrot feather. We figured out the cocktail to do that. Meaning it was a component of our product, it was a component of the other product. I think parrot feather has a nitrogen fixating bacteria in the base of it. So we kind of wipe that out and then the whole thing goes away. We've had really good success with that. This is hydrodiction. We figured this out for the customer. It took a mixture of chemistry and enzymes. He kept killing it. A week later, it would come back. So we had to stop that cycle. I don't remember where this one. I think we're showing the leaves. They had a lot of leaves there, and we put a product, we put them into pellets, we put that down. They were not oak leaves. Another one on algae, we just figured out what it was and then treated it. No chemistry, we didn't use any chemistry on this. No chemistry on this one either. This is down in Florida, this is in Tampa Bay, Florida. One of our companies, about 3 years ago, they said, you know, you bug guys, we hear about 100 of you every day. You know, I need to hear from another one. So we gave them some product, and they went from about 4 feet of sludge and every weed imaginable to about 3 feet of open water at the top. They used a muck degrading pellet we make and a product, an enzymatic product called Ponzilla. Another one in Texas. I'm getting a big one. This is one in Northern California by John there. Okay, this is a big one right here. This was this summer. This guy's on a golf course, so we couldn't use any chemistry. This is all dead hydrilla and naiad. He put peroxide on there and he killed about half of it. And he's like, I have a problem. You know, I need to get rid of this. I got a big golf tournament coming up. They got all the PGA tournaments and all this. And he said, I'm not having time to get my guys raking it off. And we know the chemistry of those weeds really well. So I said, we can break those down. We put the pellets, we have a product called Ponzilla, we sprayed that over the surface. This was 2 weeks later. And that's no BS. We've done that probably 10 or 20 places around the country, degrading that. So killing it isn't enough. After it's dead, put something in there. It's not just our product. Our product triggers natural selection. Natural selection starts to take off the phytoplankton, the zooplankton, everything, and then you'll start to get rid of stuff. I don't remember what that one was. Here's some cattails that we're experimenting with. You can't see the results, but we're kind of experimenting with our different enzymes to make them fall apart. Anyway, that's us. If you have any questions, I'll be around. Any other things? Yep.
I have a question. It looks, the pictures are impressive. The question I have is, if you're treating a plant, how are you skirting around NPDS issues and any kind of regulator that it's not a herbicide?
We talk about degrading the plant and not killing it. So we talk about applicators being good at killing it, and we're good at degrading it. We see that really as our role. There's some plants that are going to disappear if we start to use our product, but we just talk about being able to degrade it after it's dead.
Another concept you could possibly think about using would be interrupting its life cycle.
Yeah, yeah. And that's exactly what they're doing on a bunch of that.
Interruption, disruption, naturally, you know.
Yeah.
Could you explain what exactly you mean by degrading as opposed to killing, and how it affects the DO levels in the pond over time?
If you can take a plant and wound it, and then you find a way to get our bacteria or a naturally growing bacteria to look at that plant as a substrate, then it'll degrade it and take it away. So you're taking that plant, and in some way you're turning it into just a complex mixture of proteins and carbohydrates that's easily degradable. You're turning it into cotton candy, so to speak. You're making it available food stuff. And stuff like hydrilla, that grows really fast. It's really susceptible to attack because it grows so fast. Stuff like eelgrass that has a thick coating on it, that's very tough to get at. So we're trying to find ways to penetrate it, or to get the algae side to penetrate, but then to get it to degrade, make it into a food source.